Hi everyone. In this module, we're going to be talking a little bit more about the structure of DNA, and we're also going to learn about how DNA replicates. We will also talk about chromosomes in this particular module as well. But let's go ahead and start off with DNA. We're going to do a really brief history of the discovery of DNA and how we figured out not only what the molecule looked like, but also how we figured out that DNA was the hereditary molecule and that it wasn't some other type of molecule that we have in our cells. Then we're going to go through DNA structure, and I'll cover that in a little bit greater detail than I'm going to the history of DNA, and we'll also talk about how DNA makes copies of itself. So for these first few slides with the history of DNA, I'm going to kind of go through this material fairly quickly, not because it's not important, because it certainly is important, but just it's not incredibly important that you remember every single discovery along the way to the path of finding DNA, right? So this is kind of just like a brief overview. So if I kind of skip over things or go through things quickly, again, don't worry about it. It's nothing that you will be held responsible for as far as what you'll need for a quiz or an exam. So again, these are just a general overview of discovery of DNA. So there was a Swiss physician and biochemist named Miescher in 1871, and he was the first person to isolate a white blood cell nuclei. So remember, we talked about cell structure, and we talked about the nucleus and how we have all of our genetic materials inside of our nucleus. So he was the first one who actually isolated a single nuclei. He was the first person to do this. And what he found inside of the nucleus, through some chemical tests that he did, is he found there was an acid substance inside and that there were nitrogen and phosphorus present in the molecule. So he called the molecule nuclein, okay, is what he first started, because again, it came from a nuclei. So he said it was a material that was found in nuclein. Later, we specifically called it a nucleic acid. Again, the word nucleus and nucleic acid kind of go along there, meaning within the nucleus, right? So then we have not a whole lot happening for a period of about 50 years. And then Frederick Griffiths was a microbiologist, and he was working with bacteria, different types of pneumonia bacteria. And there were two different types. There was a bacteria that had basically a smooth outer covering and a bacteria that had a rough outer covering. So one was called type S and one was called type R. Interestingly enough, the smooth one was the one that was very virulent, which that means it infects people quickly, and people are more susceptible to be infected and potentially even die from that type of bacteria. So the more virulent a bacteria or a virus is, the sicker it can make you. So here's what he did. He figured out that something in the bacteria was being replicated inside of the living specimen that he was injecting it into. And he wanted to figure out what it was. So here's what he did. He had all of these mice, and he injected them with different types of bacteria. So the first one he injected with was the type R bacteria. That's the one that doesn't have a capsule, and it's not virulent. It doesn't make anybody sick. So he injected the mouse, and the mouse was healthy. Then, just to test it, he also did it with type S, and the mouse died because, again, it's a virulent strain that makes you very sick. So then he took the virulent strain, the heat-killed virulent strain, and he heated it up, okay? And then he injected it into, and he found that the mouse was still alive. But if he took the heat-killed virulent type of bacteria and he combined it with rough non-virulent type bacteria, the mouse still died. And interestingly enough, he found those live virulent bacteria cells inside of the mouse, even though that's not what he put in the bacteria. So he figured out that there was something in the bacteria, even when you heat it up, which was basically killing the proteins in the bacteria, there was something else in the bacteria that was still alive, that was still making copies of itself, which made that mouse die. So then we have some physicians who kind of did something similar, basically, and they came up with this idea that DNA, they kind of explained what Griffiths did, DNA was what's responsible for converting those non-virulent R-type bacteria into the virulent S-type bacteria, because the DNA was still present in the S-type even though the protein itself had been killed. So now we move on to Hershey and Chase, and they wanted to figure out, again, is it bacteria, or excuse me, is it DNA or is it proteins that are actually responsible for transmitting hereditary information? So they did something similar, but they did it with viruses instead. Okay, so very similar to what we saw with Frederick's doing with the bacteria and the mice. Hershey and Chase just did the same thing, but they did it with viruses instead, viruses that infect bacteria. And without going through this in a great amount of detail, the moral of the story is DNA is the hereditary molecule and not proteins. Okay, proteins aren't hereditary molecules. They don't transmit genetic information. DNA does. I'm going to skip over this one very quickly, but this was Levine. He was a Russian-American biochemist, and he basically discovered there were 3 parts to the nucleotides that were found in equal portions in a DNA molecule. It was a sugar, a phosphate, and a base, which we're going to talk about more later. Chargaff figured out some information about the types of bases that are in DNA, but again, I'm going to skip over this right now, so don't worry about it. And then we've got some English scientists, Rosalind Franklin and Maurice Wilkins. So they figured out there were kind of 2 forms of DNA, and they used this technique called X-ray diffraction to kind of take a picture of DNA. And why this was so important was because we knew, okay, DNA is a hereditary molecule, fine. We know that it consists of these smaller subunits called nucleotides, and the nucleotides are made up of sugars and phosphates and bases. But what no one could figure out is what it actually looked like. We couldn't figure out the structure itself of DNA. And so Rosalind Franklin's work was incredibly valuable because she came up with a picture using X-ray crystallography, and this is what this picture looked like. So as it turns out, 2 other scientists, Watson and Crick, they got their hands on a whole bunch of information. They didn't do any experiments themselves. They didn't actually discover anything other than putting all of the pieces together that everybody else had figured out. So they took what Chargaff had figured out, and they took what Levine had figured out, and what Wilkins and Rosalind Franklin had worked on, and they put all of the information together to actually come up with the model of DNA. Okay, so again, here it is. This is James Watson and Francis Crick and sort of their famous model of DNA. So they are given credit for discovering the structure of DNA, even though it was the work of many other scientists that actually went into it. So here's kind of a nice table from your textbook that summarizes all of the different contributions, again, of the different scientists when it comes to the structure of DNA. Not necessarily something you have to memorize. Again, you should probably know Watson and Crick and even maybe Rosalind Franklin because of their the importance of what they put together. But again, it's not necessary that you know all of these individuals and a great amount of detail of their work. But the moral of that story is, yay, we figured out what DNA looked like. So now what we need to do is let's learn about what DNA looks like ourselves. So I've mentioned a couple of times that DNA is assembled from building blocks. And those building blocks are called nucleotides. And the nucleotides contain 3 things: a deoxyribose sugar, a phosphate group, and a nitrogen base. So deoxyribose sugar is how DNA gets its name. So DNA stands for deoxyribonucleic acid, right? So deoxyribo comes from the type of sugar that we're using. This is an important distinction because in another chapter, we're going to learn about a different type of sugar and a different type of nucleic acid called RNA. So it's important to know that there are differences. So the nitrogen bases that can be attached to the sugar and the phosphate are either adenine, guanine, cytosine, or thymine. And we classify them differently based on the number of rings they have. So adenine and guanine are purines, cytosine and thymine are pyrimidines, and purines have 2 rings, and pyrimidines have 1 ring. So that's kind of the only difference between them. So here we have what would be an intact nucleotide. So you can see this sugar here, the deoxyribose, which is attached to a phosphate group. And then we have these nitrogen bases. In this case, we're looking at guanine. Okay. What is important to know is that DNA, excuse me, is a double-stranded molecule. So we have all of these nucleotides that are being attached in a backbone with sugar, alternating sugars and phosphates. Okay. And then into the middle, we are sort of projecting these nitrogen bases. So we simplify them and just call them A, C's, T's, and G's for shorthand. So we've got, again, a continuous sugar phosphate, what we call backbone. And then in the middle, what we're going to do is we're going to take another single chain of these nucleotides, and we're going to attach them. So we're going to attach two of these chains together. So once again, the nitrogen bases are interior, and they are going to actually bond with each other, which just means they're going to hold very weak associations with each other so that they kind of hold on with each other. And it forms what's called the double helix. But, excuse me, pardon me. What's interesting to note here is that the strands are not parallel. They're actually anti-parallel. And you can kind of think of this as if you take a hand and then flip the other hand sort of upside down and stick them next to each other, they would almost look like mirror images of each other, right? Okay, so they're not running parallel, they're running anti-parallel to each other. That only becomes important in our discussion of replication, DNA replication, which we'll talk about in just a moment. So I'm going to kind of skip over this part about 5' and 3' sugars. It's not 100% necessary that you know the 5' and 3' carbons, and it doesn't really matter for our discussion. All you need to know is that one strand of the DNA molecule runs one way, and the other strand of the DNA molecule runs the opposite way. Okay, so however you want to think of that, again, they're anti-parallel. So how do we know how these strands are going to come together? Well, it actually turns out that there's a very specific way that those nitrogen bases pair up together. Purines and pyrimidines match each other. So remember, purines had 2 rings and pyrimidines had 1 ring. As it turns out, adenine and guanine, cytosine and thymine. So we have adenine and thymine always make bonds. See, the double ring and the single ring. And guanine and cytosine always make bonds. Again, the double ring and the single ring. So A's go with T's and C's go with G's. Okay, that's kind of the universal rule of base pairing when it comes to DNA. A's goes with T's and C's goes with G's. Always, always, always. So what this means is that when we are looking at DNA sequences, and what a DNA sequence is the actual order of the letters. And the order of the letters is a code that's going to tell us something, and I'll share what that is in our next lecture. It's a code that tells us something. And what's interesting is if we only have half of the message, if we just have one side of the DNA molecule, that's fine. We don't need the other side because we automatically know what's on the other side because of these complementary base pairing rules. So we don't need to sequence both sides of a DNA molecule when we're sequencing genomes. We only need to sequence half of it because we already know what's on the other side because of these universal rules. Okay, let's do a quick comprehension check. In DNA, the amount of an adenine is always equal to the amount of what? Ribose, deoxyribose, thymine, or guanine. So remember, only thymine and guanine are bases, T's and G's. And it's actually thymine, right? A's go with T's and C's go with G's. So the amount is always equal because we're going to have just as many A's as we're going to have T's and just as many C's as we're going to have G's. All right, so DNA, we kind of talked about this a little bit, what DNA looks like inside the nucleus in its resting state and what it looks like in its very condensed chromosome state. So we kind of talked to this a little bit before, but what's interesting is that we take these individual strands of DNA and we sort of wrap them up on something called histones. It forms what's referred to as nucleosomes, and you can kind of think of this as being like a beaded necklace. Okay, so if you think of a beaded necklace and the beads represent these histones, and then the DNA will wrap around those histones and it'll form sort of this long beaded chain. And then what we can do with that beaded chain is we can coil it up, and that kind of forms our chromatin. And then when we want to make chromosomes, we will then squish all of those loops together to form chromosomes. So again, it kind of looks a little bit like this. So here's our naked DNA, okay, but actually inside of our nucleus we have these histones. And once you wrap DNA around the histones, it becomes a nucleosome. And then you can wind it up, coil it up, and turn it into chromatin. And then in order to make chromosomes, you can fold those loops back and forth, back and forth, back and forth, and make these really tight chromosomes. So it's very compact, the way that we're fitting all of this genetic information into this itty-bitty nucleus we have inside of our cells. All right, so if you guys remember, we have genes, which are specific segments of our DNA that tell us that we're going to make a protein. And then we have the proteins, which are actually, again, the molecular, so again, they create this phenotype, right, a trait. So there are genes for all of our physical traits. There are genes for every function in your body that you can possibly imagine. And once again, that information is conveyed by the sequence of those nucleotides. So we read those letters, and that code tells us what protein we're going to make. We're going to talk more about proteins in, again, the next lecture. So before that, let's talk about how DNA replicates. So this is an important concept. So if you guys remember from our discussion in Chapter 2 and even Chapter 3, when we were talking about mitosis and meiosis, in the S phase of the cell cycle, before we can divide, we've got to make copies of everything. And this is very fast. We can do this at about 50 bases a second. That's how quickly your cells can replicate DNA. The human chromosomes, so remember we have 46 chromosomes, 23 pairs, we have to do this simultaneously at hundreds of different points along the length of the chromosome. So this process is incredibly complex and complicated. And we start off at these places called replication forks. So what we do is we unwind the DNA, so we separate the two parent strands, and then once the parent strands are open, what we can do then is begin matching up free nucleotides. So our body has created a bunch of free-floating nucleotides that are just waiting to pair up with a complementary base pair, okay? And then we pair up all of those free nucleotides according to our base pair rules. And then the sugar phosphate backbone, okay, comes together of our new, what we're calling daughter strand. So that's the strand that was replicated using the original strand as the template. So these are the enzymes involved. I'm not going to go into that in great detail. There's a bunch of proteins that help this process happen. They unwind the DNA, they bind things, they read segments, they put in the correct free-floating nucleotides, they join the backbone together. But again, we're not going to go into it in great detail. But here's what it would look like. We would unwind it. Our primase is going to start reading the sequence, and we're going to start putting nucleotides on one after the other, okay? And it's happening on both strands. Again, these are the parent strands, so these are the template that we're using. And then again, we're creating these new strands, which are our daughter strands. And we synthesize on one side. We can actually synthesize continuously because it's the right way to read our DNA. On the other side, we can't because it's backwards and we can't read backwards. Just like if you're reading a book, you can read left to right, but it's very unlikely you know how to read right to left. It's just something we don't do. That doesn't make sense to us. Same thing with our DNA. Our DNA can't do that either. So it creates these little small fragments that are eventually bound together. And then the primases come off and we bind everything up. And ta-da, that's replication. Okay, so quick comprehension check as we finish here. When DNA is replicated, each daughter molecule contains, so this is new DNA, one parental strand and one daughter strand, either two parental strands or two daughter strands, a single daughter strand only, or two daughter strands only. The parental strands are destroyed. So when we're all finished with DNA replication, our new molecule of DNA contains which of the following? It's number one. It's one parental strand and one daughter strand, right? Because we've got original template that we used, and then we've got the new strand that was synthesized. All right, so these are the important terms and concepts for Chapter 9. Again, don't worry too much about the history of DNA. There's a few important points, but don't stress over it by any means.